I'm going to talk today about the impact that Jim Simons had on neuroscience because I think it is so substantial and such an exciting topic. And I'm going to start with a little piece of personal history. So in 2001, the National Institutes of Neurological Diseases and Stroke had a symposium on its 50th anniversary, and I gave a talk there about the work my lab was doing on the genetics of social behavior and how different individual worms would be either social or solitary in their behavior patterns, and how the genetic basis of that worked and what happened. And after that talk, two women came to talk to me, and they were from the National Alliance for Autism Research, which was a patient organization founded in 1994, later became part of Autism Speaks. And they said, you know, we think autism has a biological basis. Your work on social behavior could help us learn more about the biology behind autism. You should think about that. And I was completely stunned because at that time, the experts on autism were special education teachers in elementary schools. And the idea that genetic studies in a worm could shed light on anything that was going on with human children was completely out of the box. I would never have put that into a grant application. But I did think about it, and I actually had a childhood friend named Kathleen Kennan, who was a special ed teacher. And I asked her about it, and she said, you know, autism is really interesting. You have kids come in with learning disabilities, and for some of them, like Down syndrome kids, you know exactly what their future can hold. You know what their capabilities will be, what they will and won't be able to do when they're 18, where they'll need special help, whether they can live independently, where they'll be challenged. But if you have two nonverbal kids come in along the autism spectrum in kindergarten, they look really similar. You have no idea where they'll be later. One of them will maybe never speak, and the other one will maybe show up in junior high school completely normal, maybe playing a lot of computer games. And you just work to find the right approach. It's very personal, and you uncover their potential. And I still think of that conversation because it perfectly captures what we've learned since then in 25 years of science of autism. And that is that it's extremely heterogeneous. It's many different things. And so it's not one particular outcome. So 2001, science was very weak in this area. So child development was studied by psychologists, not by biologists. The mid-20th century was when autism was first described, and I am really sorry to say that the cause was proposed to be the refrigerator parent, a disorder of parenting that led to the child becoming socially withdrawn. And evidence against this theory began to develop in the late 1970s, when people came to realize that identical twins had a much higher propensity to be both affected with autism than non-identical twins, which indicated there was some kind of a genetic component. But it was decades before we got to the bottom of that. And it was also clear that there were other things going on, not all of them genetic, and it was very hard to sort of trace down what the environmental and genetic factors might be. And in the meantime, there were other false starts, including a now very much debunked paper in 1988 that suggested that autism was, as well as colitis, was caused by the vaccine against measles, mumps, and rubella. It was just nowhere. And this is Jim Simons enters, okay? And in 2003, Jim asked a pretty simple question: how is it possible that we don't know anything about autism? And taking the approach that you've already heard about, he decided he wanted to discover this, and he wanted to turn this psychology, this child development field, into a science. And he's pursued the basic science in just an amazing way. So in 2003, Jim and Marilyn convened my Rockefeller colleague, Paul Greengard, Tom Insel, who was then director of the National Institutes of Mental Health, a number of other scientists, and they just discussed what it would take to make scientific progress. And this was very much Jim's, like, let's just have the smartest people in the world in the room and talk about the problem. And they did, and they agreed that what they should do is to follow up on that clue from the genetics. There had to be something going on with those identical twins. Let's make some progress on that. And they also said, you know, once we can find some genes, then we can study the neuroscience, then we can study the brains, and then we'll be able to make some progress. And it was a good time to do that. The first draft of the human genome had come out, actually was announced 25 years ago yesterday, so that was just coming up, just coming online. And Jim and Marilyn decided to get started. They made grants to Rockefeller, Yale, MIT, and very importantly, Mike Wigler at Cold Spring Harbor. And Mike Wigler is here with us today to start to study this work. And within just a couple of years, that work really led to a breakthrough in our understanding of the genetics of autism. And that was work that Mike Wigler and Jonathan Sabat published in 2007, titled Strong Association of De Novo Copy Number Mutations with Autism. And so what they showed was that in some autism patients, some children, there were small either deletions or duplications in their genome that were not present in either of their parents. And they would sometimes take out a couple of hundred or kilobases of DNA, one gene or multiple genes in a region. And this kind of helped explain something about how you could have something that was genetic but seemed to appear out of nowhere. It wasn't in either parent. It was new. It was in the sperm or the egg of that had given rise to that child. And it led to an amazing sort of understanding. It actually opened the door to a huge amount of understanding about certain things in the genetics of the human brain and of human behavior. So most genes in the human genome, you have two copies, one from the mother, one from the father, and that's how it works. But sometimes you end up with one copy or three copies, and most of the time that's perfectly fine. And those of us who have studied model animals, most of the time we know there's no problem if you just have one copy of a gene. The system is robust. It can get around that. But it turns out that the human brain has evolved very special capabilities, very special cognitive capabilities, and this ability of communication that we have with each other. That is our superpower. We talked about mathematics, right? So in the 17th century, Newton and Leibniz independently developed the idea of calculus. And no one in the history of mankind had ever thought of calculus before, and as far as I know, no animal has ever thought of it either. But now we teach it in high school, and everyone learns it. It's amazing that we have this ability to communicate with each other. It's an astonishing ability, but it's very recently evolved, and it's very delicate. And so it's fragile in ways that other aspects of our fundamental biology are not. And so small changes, like even just having half as much of a gene or 1.5 times as much of that gene active in different tissues, can lead the brain to be unable to do this incredibly difficult task that we have set for ourselves. After a couple million years of evolution, I'm sure it will be much more robust. But for now, we see this in this kind of special vulnerability that these children have. Around the same time, a couple of other groups started finding specific genes that were mutated in very rare cases of autism. Thomas Bourgeron in Paris found a couple of genes that were well understood to be involved in synapse formation, the formation of connections between different neurons in the brain. Neuroligands and neuroligin 3 and 4 and Schenck. And that, too, gave us a really important clue because that said, okay, we should be thinking about the development of the brain. We should be thinking about the connections between neurons. That is something where someplace is happening. So these things were just electrifying. All of a sudden, we had a way of studying these problems. But it was still really, really difficult because the fact is that the things that Mike Wigler discovered, these copy number variations, they were very rare, and each individual one was very, very rare. And the neuroligin mutations were very, very rare, even in people with autism. And so no one of these things could explain more than about 1% of what was going on. So we had to look much more broadly. And that was something where, this is where Jim and Marilyn really stepped in and made a difference, because they realized that people were going to have to work together. This was a big problem. And maybe coming from physics, where people work on big problems together, was something that sort of guided them to start to think about doing that in biology and in neuroscience as well. I don't know. But when something important is happening, people start to gossip about it. And from an email around that time, I heard, here's something. Jim Simons is setting up a whole autism foundation. He's a hedge fund guy, supposed to be smart, and apparently reads the grants himself. I learned later that not only does he read the grants himself, but he would actually come to all the talks and sit in the front row, as we heard about. He's incredibly interested and passionate and committed to the problem. And in 2006, he recruited Jerry Fischbach, we're in the Jerry Fischbach Auditorium, to run what became now the Simons Foundation Autism Research Initiative, or SAFARI, with the mission of pursuing the genetics, bringing in relevant neuroscience, like those developing synapses, and elevating the field. And they recruited outstanding scientists to work on this. And I know some of the people in this room are really interested in philanthropy and how you go about they created a new problem in a new field. And they didn't do that by sort of taking the people who were already out there. They did that by really bringing in a whole set of new people with new approaches and new ideas. And so, for example, they brought in Tom Südhoff, who worked on synapses. And he later won a Nobel Prize for that work. And bringing people like that into the field kind of made it safe for other people to come to the field, And it sort of made young people feel like, hey, there's going to be something here. I can build a career here. And it's also, they also had this attitude, like, you don't have to have any experience in this field. You don't have to have preliminary results. You just need to be bold and imaginative and smart and rigorous and willing to learn and willing to work with other people to get this to happen. And that was the whole goal of what the Simons Foundation did in autism. Is very cooperative, very much about creating the extra intelligence it has from having people work together. And that is what Jerry and later Lou Reichard and now Kelsey Martin have been doing in the neuroscience program at the Simons Foundation. And I can just very briefly say that starting with, they taught us so much about genetics and neuroscience. In principle, it was sort of, it was basic science that addressed areas that had not been addressed before. So I mentioned this thing about the vulnerability of the brain to copy number variations. They taught us, they used animal models and studied social behavior and looked at synaptic remodeling and sex differences in the brain and technologies. And every time they're sort of thinking about the basic science of autism, but they were kind of bringing whole new areas that are just really important. Because the fact is that autism is so closely allied to what we are as human beings that by studying it, you're studying these profoundly important questions about brains and behavior and humans. So a lot of really impactful things came out of that, including, for example, working with different patient groups. They've recruited tens of thousands of patients who've had their genome sequenced, so we now know a lot about the underlying genetic basis. There are indeed hundreds of genes that can contribute to autism and related disorders. They've also worked with families and created, for example, groups that bring together families that all share a particular genetic change. And it turns out that if you have a particular genetic change, the kids within those families might have the same strengths and weaknesses, and it provides a real support group to be working with other families and figure out what you can do to help those kids out. And they also, so then from the animal models, they kind of pulled up a couple of other threads, like where are these genes, when are they active, where are they important? A lot of them turn out to be active late in pregnancy when the large complex circuits of the brain are first forming. And again, I mentioned vulnerability. Billions of neurons in the human brain are all trying to form connections with the right billions of neurons at that time. And you can start to look at subsets of those genes and realize what they're doing at different times in development. So for example, some of them are about synchronizing the development of different classes of neurons that are supposed to be connecting at the same time. And if they get a little bit out of time with each other, the connections get a little bit muddled. Not dramatically, you're not uncoordinated or in other ways like dramatically neurally affected, but this idea of developmental synchrony and timing of the connections turns out to be important for one aspect of autism. And then there were also animal models that enabled people to kind of look at different kinds of pathways that are altered in autism. And I have kind of a favorite story that involves a human to animal to human connection. And that is that many people with autism report having sort of hypersensitivity to sensory stimuli and just being unable to filter out all the information that comes from the outside. And Temple Grandin, who's a woman with autism who completed a PhD in animal science and is a very successful professional and then wrote about her experiences, talks about this. She says, you think my problem is that I don't want to communicate with you. I think my problem is that I am completely overwhelmed by stimuli that I cannot filter out. Maybe if you help me with my problem, I'll help you with yours. And about 10 years ago, Lauren Orefice and David Ginty, founded by Safari, showed that mice that have genetic changes mimicking some kinds of autism are hypersensitive to touch stimuli, and that they have certain kinds of anxiety and activity patterns that are associated with that hypersensitivity because they cannot filter out that excess stimulation. And so these mice have, the social interaction deficits have that same kind of element that you would have that Temple Grandin reports. And then you can start to make threads between even some of the completely, apparently unconnected ideas that were relevant to autism. So one of the other factors that we realize is important in autism is that during pregnancy, again, there's this time that the brain is developing that it's kind of fragile. And if the mothers are infected, maternal infection, during that period can result in a higher incidence of autism and related disorders in the children. And Gloria Choi's lab at MIT, another group that's been supported by Safari for a long time, has shown that maternal immune activation in pregnant mice leads to social deficits in their pups. And because you're doing this in mice, she was able to trace it down, like what is it that's happened at this point? And it turns out to be some of the areas that are processing sensory stimuli, exactly those same kinds of touch and auditory stimuli that are observed to be abnormal or the sort of overwhelming to patients with autism. So here you have like a completely different causal factor, this immune activation, but you start to converge on some of the related ideas about touch and sensory processing in certain regions of the brain and certain times of development, and all of them related together. And all of this is, again, not just relevant to autism. It's incredibly interesting to me as a neuroscientist, and it's relevant to autism. And it helps us think about a subset of these individuals in a different way, and perhaps a refreshing way that we can use to make progress. So, just tremendous. I just want to say that the Simons Foundation has also supported another project very close to my heart, which is the nature of thought. And this is, again, Jim decided about 10 years ago that he was ready to ask another question, and it was a big question, and he decided that his favorite pastime was, I guess, lying on the bed looking at the ceiling. We just heard from Marilyn, was thinking. And he wanted to know what was going on in his brain while he reflected on a math problem or developed a new method or learned a new skill. And neuroscience is a field where people were generally studying one neuron at a time or one brain region at a time. But the defining function of the brain is the involvement of billions of neurons. And that's what you're doing when you're thinking. You're kind of drawing from all of these different resources, from memory, from past experience. And it was hard to understand how to put that together. We could look across the whole brain with these kind of slow methods like fMRI, not super helpful for high resolution. We could look at individual neurons, but then always very specifically. And Jim said, this seems like another big problem, where we need a lot of different people working together and thinking across fields and doing something interesting. And he found a few neuroscientists who are thinking the same way. And David Tank, a physicist turned neuroscientist, and Tony Movshan, a sort of cognitive neuroscientist with a lot of experience in theory, led this Simons collaboration for the global brain, which had an amazing 10-year trajectory of moving from this sort of one region at a time to really thinking about the brain as a whole over the work. And this was something where, new tools were becoming available, so you could look at many neurons at a time. But it was also just, what can you do? You could look at many neurons, you could manipulate their activity. You had to build the math and the quantitative methods and the theory to interpret what you were going to get out of looking at tens of thousands of neurons at a time. They put all these people together, and they worked together looked for common themes. They were working in all kinds of different brains and different systems, but then they would kind of combine their information in these very Simons-like meetings, pulling together people with different collaborations. And it's made amazing progress toward understanding what thinking is. And I'm just going to sort of say that the formulation when they went in of how the brain works was almost like an assembly line. You know, you do this here, and then you give it to the next person in the line, and then you take it to the next person, and then to the next person. And they really showed that that is not how the brain works, that it's really global, that it's broadcasting information and cycling it back and forth all the time. And in fact, the information is being transformed. So you might look at the same part of the brain and the same neurons at 2 different times in the same task, and they will have switched their properties. Okay? So there's something about the internal state of bringing in this new information that's remapping that information. And because of the ways that they were doing this, they're able to actually recognize internal calculations from the activity of the brain, because they had made all these measurements and done all these perturbations, and they knew what the brain was doing in these different times. And they're really starting to define the nature and the role and the mechanisms of the neural activity that are associated with cognition. This is really exciting work. So they say that imitation is the sincerest, so that's the science, which I just find compelling and amazing and really inspired by Jim's example. And they say that imitation is the sincerest form of flattery. And I want to say that when I was asked by Mark Zuckerberg and Priscilla Chan to start a philanthropy in science about 9 years ago, my first operations manager and I came to visit the Simons Foundation in New York, and Marianne Greenup and Marilyn Simons very graciously sat down with us and helped us talk about what to do. Marilyn's also been something of a mentor to Priscilla Chan in her work in philanthropy. And we just had copied what Jim and Marilyn did. We looked for big ideas, we listened to the experts and the smartest people we could find. We tried to do things that other people thought were too ambitious or that were too new. And we brought together people from experimental science and medicine and from computational fields, kind of bringing the math and the biology together. And we asked people to work together and to share their results. And this is all really, I think this is a great model, again, for those in the room who are thinking about philanthropy. So I want to close on a personal level. Jim Simons, great taste in people, asked big questions, learn from the experts, intense engagement. And you know, he didn't have to be the smartest person in the room, even though he was. You know, he was always, he listened. He was great. And to me, Jim is like, he reminds me of the Ancient Mariner in the Coleridge-Taylor poem, The Rime of the Ancient Mariner. So just for those of you, just a reminder, this is a poem that starts with an ancient mariner, and he stops this callow young man on the street who's on his way to a wedding. So he holds him with his skinny hand. There was a ship, quoth he. Hold off, unhand me, grey-beard loon. Eftsoons his hand drops he. He holds him with his glittering eye. The wedding guest stood still and listens like a 3 years child. The mariner hath his will. So the mariner hath his will means that the mariner proceeds to tell 159 verses of a story while the wedding guest listens. And it's a very good story. It involves an albatross. So why does this remind me of Jim Simons? First of all, of course, the mariner, the ship. Jim had a boat. He took it to wonderful places, took people along. I went along on one of these boat trips at one point because he would invite people he thought were interesting. Mike Wigler was there. Audrey was there as well. There was not an albatross, but there was an incident involving a pelican. So there's a boat. Okay, second reason, like the Ancient Mariner, Jim was a great storyteller. So some of these stories, any of you who've sort of been there when he was just a raconteur and someone who would go on, you could listen. I don't know if it was 159 verses, but he was really someone who shared with people as well. Third reason, he holds him with his glittering eye. Jim was compelling. He would just bring you in, and you would just have to go where he was going. And I think that was true. And then the fourth reason is like the Ancient Mariner. By doing so, Jim did the right thing, but he also persuaded others to do the right thing. And that's the end of the Ancient Mariner. So he showed by his example that philanthropy is a privilege and a joy. Thank you.